Hello everyone, my name is Tim from the School of Medicine, Physical and Rehabilitation Department. Today I would like to share with you a novel approach toward treating blindness. Hopefully at the end of my presentation, you're able to learn something new about the visual system. Macular degeneration and retinitis pigmentosa are the two infamous retinal degeneration disorders characterized by the loss of the photoreceptor layer that eventually leads to permanent blindness. So this is a normal retinal structure, and in case of retinal degeneration, you lose this layer of photoreceptor cells. Studies have shown that even though this layer is deteriorated, other layers are conserved, especially the ganglion cells layer where the optic nerve is. And this gives us a hope that replacing the photoreceptor cells might help rescue the retina and possibly reverse blindness. So how do we do this? We use something called the human embryonic stem cells, which is the blue inner cell mass obtained from the developing embryo. We add a lot of potions containing neurotrophic factors to differentiate them into something called the retinal organoid, which contains developing retinal layers. After that, a portion of this retinal organoid is transplanted into the glass retina and utilizes the host transcription factors to differentiate them into retinal components, such as cones and rod photoreceptors. So we grow these cells and transplant them into the suprarenal space. OCT is used to monitor growth in vivo, and after 4 months, we see these silver dots as a good sign that the transplant is alive and thriving in the host retina. If you don't believe your eyes, we can color in the black and white images to show you that the dots are actually clumps of cells called rosettes and containing differentiated cells. We then use immunofluorescence assay to examine what cells there are. We use KUAD to visualize transplanted cells in green. We also use RHO to stain rhodopsin, which is a protein found on the rod photoreceptors, in red. We can visually detect the colocalization of the two stains, suggesting that the transplanted cells have successfully differentiated into rod photoreceptors. Besides rods, we attempted PKCFF for rod bipolar cells, recovering for both rod and bipolar cells, and CrowdBP1 for mucoclia. And as you can see, these samples all yield positive results for these antibody markers, suggesting that these cells are present in the suprarenal space. Moreover, when staining with KUAD, which is a transplant marker, it shows colocalization of the two markers. This suggests that the transplanted cells are able to differentiate into photoreceptors as well as important retinal specialized cells. So in conclusion, the study shows that the transplant survives and reaches maturity in the host subretinal environment and appear well integrated with the host retinal architecture. Moreover, these cells are powerful enough to give rise to even more specialized cells that are important to establish a functional retinal circuitry. Thank you for listening in and hope there was an eye-opening talk.